Hello everyone, I hope you've been staying safe over the Easter holidays. We'll get back to it now. I'm going to start today with an overall explanation of the events that take place during meiosis. Now, there is more detail that you could go into, like the stages of the cell division, but I'm not going to spend too much time on that to begin with. We'll look at that later when we look at how genetic diversity is increased through meiosis. And those stages are similar, if not exactly the same, to the stages that take place during mitosis from previous lessons. So, this diagram is in the notes, or a version of this diagram at least. It shows an adult somatic cell here. This is a diploid cell. It has one paternal and one maternal chromosome. So on here I'm just going to use the annotation. So the dark ones are the paternal chromosomes, and these are the maternal chromosomes. So paternal inherited from the father, maternal from the mother. Now, I have explained on a previous video about the way to count the chromosomes, and also Mrs. Andrews has done a really useful explanation of counting chromosomes as well. That is one of the major areas where people get confused when looking at meiosis. So we'll try and cover that a little bit again today. So this cell here is diploid. Diploid means it has the homologous chromosomes. So these are homologous chromosomes. If we remember that homologous chromosomes are, or homologous pair, are two chromosomes with the same genes at the same loci. Okay, so homologous pair, two chromosomes. Homologous means they have the same genes at the same loci. So if we just imagine that, we've got a gene here. The same gene will be present on the paternal chromosome as the maternal chromosome. Now, it might be that the alleles for those genes are different, but that's not important. This is the definition for homologous chromosomes. So, in a diploid cell, the homologous chromosomes are present. Between this stage and the beginning of this bit down here, which is actually where meiosis is taking place, where the cell division is taking place, these chromosomes undergo DNA replication. So if you need a reminder of that, I've done a video on semi-conservative replication. You can have a look back through there. The two strands in the DNA molecule become separated. 3 DNA nucleotides line up opposite their complementary base pairs, and then DNA polymerase forms a phosphodiester bonds between those nucleotides to form two new DNA molecules, which each contain one original template strand and one newly synthesized strand. Like I say, go back and have a look at the DNA replication video if you need to. So, between this cell and this cell, DNA replication has taken place. This is where we come to this idea that sometimes people get a bit confused with the counting. Sorry, my board's rattling, a bit of noise. With the counting of the chromosomes. After DNA replication, this cell is also diploid. It still contains homologous pairs of chromosomes. So here is the paternal chromosome, here is the maternal chromosome, still existing in their pairs. The only difference now is that the DNA has been replicated. So that strand, or that molecule, is replicated to make an identical copy, and that's shown there. Sometimes questions might ask you to explain the appearance. God, that's really annoying, that rattle. I'm sorry about that. To explain the appearance of one of the chromosomes here. So what we would say are there are two sister chromatids. So each one of these is a chromatid held by a centromere, and that's the centromere here holding those two sister chromatids together, and the centromere holding those two sister chromatids together. And those two sister chromatids are the product of DNA replication. So what we're saying here is this molecule of DNA is identical, the nucleotide base sequence is identical to that molecule of DNA, held together by a centromere. This cell is still diploid. And just a reminder, sometimes diploid is written as 2N in a shorthand way of representing a diploid cell. Like I said, Mrs. Andrews done a really good video on explaining how to count the chromosomes. One other little technique that I sometimes use is I draw the centromeres on all of my chromosomes. So I draw a centromere there before DNA replication, and I can count then the centromeres rather than counting the molecules of DNA. So I can see 1, 2, diploid, 1, 2, diploid. So there's one homologous pair and a second homologous pair. Okay, and that gives us this diploid cell at the end of interphase, just before cell division takes place. Then we move into this first meiotic division, or you can call it meiosis 1, or the first meiotic division. And during that first meiotic division, well, we can see that the homologous chromosomes are separated. Okay, so that's the key event that takes place during meiosis 1. Homologous chromosomes, or I think it says in the notes, the members of the homologous chromosomes are separated. Okay, into two daughter cells. Now each of these daughter cells now contains just one of those members of each of the homologous pairs. So again, if we count the centromeres, or if you count the homologous pairs, we've just got one of them, and you've guessed it, after that first meiotic division, we can say that the cell is haploid, or N. Okay, so that's during meiosis 1. During meiosis 2, so the second meiotic division, this time the sister chromatids are separated. I've run out of space a little bit here. So we'll say during meiosis 2, that the sister chromatids separate or are separated. These cells are still haploid because they still have one copy of each of the chromosomes, just as these ones do. Well, this has one of each of the chromosomes from the homologous pair. This has one of each of the chromosomes from the homologous pair. It's just now they're not held together in their sister chromatids. They've been separated. Their sister chromatids are separated. If you want to, you can add centromeres to each of those, and you'll see in each of those cells there's just a single centromere. There's just a single centromere. They must all be haploid or N. These haploid cells that we have at the end, these are the gametes. So they could be sperm in males, or oocytes, or eggs in females. Sperm or eggs? That doesn't make sense, does it? Sperm or eggs. Okay, so these are the gametes. And that is an overview of the two divisions of meiosis. That's the first part of this little series of videos that I'll do on meiosis. The important parts to note: DNA replication, so that's semi-conservative replication. Here, those the homologous pairs are a product of that DNA replication. Each one of the sister chromatids within the homologous pair is an identical copy. The appearance, to explain the appearance there, two sister chromatids held by a centromere that are the product of DNA replication. That explains this fact. During the first meiotic division, homologous chromosomes are separated. That produces haploid cells, so the cells are haploid after the first meiotic division. Now that's often missed by students. They often think that it only becomes haploid at the end. But in fact, if we're counting the centromeres, we can remember there's only one centromere in each one of these cells, so therefore it must be haploid. Second meiotic division, the sister chromatids become separated. Each one of the gametes contains one chromosome from the original diploid cell. So it may contain a paternal chromosome or a maternal chromosome. And we'll stop there, and I'll come back with another video shortly. Okay.